we're doing this at the moment for niche learning, and this is the last thing I want to show. So I think it's clear, as you saw already in this spatial transcriptome data from Barbara, that cells talk to each other, and even though we disassociate them in the single-cell RNA-seq setting, we know from various papers that they are in contact, and this is actually one of the papers from 4Inter that I was presenting this morning. I won't say much now about it because it's quite technical, but the idea is that even though we have them disassociated, because of their local context, we see a remnant of that communication that we've been studying also in 4Inter in that particular expression. So can we now maybe take just a disassociated cell and predict what its local niche would look like? So this is this question that we're trying to do here. This is work by Ale, Anna, and Giovanni, where we now train such a transformer model, one of these foundation model ideas, and then have, for example, prediction for niche density, composition, and so on. We call this thing niche former. This is starting to work out. I just want to show you one thing, how this works. This all sucks up a lot of computation time and a lot of large-scale data sets. We're training now on, I think, up to 50 million cells. So it's really, like, even just like acquiring that data, let alone then composing this, was a lot of work. But it's kind of working out. And here you see this now on mouse brain data set that I should be actually acknowledging somewhere here, but I don't know if I did that. This was from colleagues from Alan Brain and Xiao Wang, who has been using this for Hummerfish for really doing a very deep stack of mouse brain, where we've been now looking into local regions of this brain and trying to predict now for this particular cell how it performs. Overall, here we see that this niche former actually does a very good job at prediction, but we can really zoom in. And for example, here, this would be, I think, vascular cells, and we see that we predict its niche quite well. If you have one vascular cell, its niche typically is vascular. Kind of makes sense, right? Because that's how blood vessels are formed. But then you look in other regions, and you see, for example, here that in hippocampus, maybe you sort of predict niche like this, and in parts it sort of goes wrong, say, but we're currently working on that. So to summarize, I think we're entering this age of not just building reference atlases, but taking these atlases, building them into models, and then sort of attaching our downstream use cases on top. And I think this is a very exciting time because if you have these models, you can answer maybe quite unexpected questions, such as not only giving jackets to the Pope or Taylor Swift, but also to ourselves. So I would give it back to you last time to show how space works, and I think then we're done.
Yes, just a few, very few slides. Actually, Fabian just showed what you can learn from spatial data. So what about spatial data in organoids? How does that look? How can we do this? And so this is really just a few slides at the end. So we have been working on setting up different spatial technologies, one of them being iterative indirect immunofluorescent imaging for eye, in this case on histological sections. For eye was originally developed in Lukas Peltmann's lab. This is actually a quite simple method. It is an immunohistochemical staining with primary secondary antibodies, but instead of just doing that once and imaging, we are staining and we have the right buffer so that the antibodies are not kind of cross-linking with the tissue, so that we can elute them again at the end and restain. So we do this in many cycles. In this case, 32 cycles, 78 stains. Not all of them work, but you can, in this case, have now a map of your tissue with a lot of proteins measured. This is data that is actually really scale crossing. You can look at the whole organoid here, day 7, 9, 11, 15, 21, 55, at the whole organoid slice, but you can zoom into nuclei and subcellular localization of proteins. It's, in that sense, really great scale crossing data. And what we do is actually we slice organoids, in this case from different time points, put them on a huge glass cover slip, put the superstructure on top, so that we have a 96 well plate with organoid slices in each well, and then actually we are staining all of them, imaging overnight, next morning starting the elution, restaining, and again imaging overnight, so every cycle is a day and a night. But this is then the data at the end, so just a zoom into one of these cortical areas, we have now all these different stains in the same tissue, in all these organoids, yes. And so, what we now want to do is not just look at individual stains. We want to actually bring this data together. We do this always with the single-cell transcriptomes. We don't look at individual genes. We actually do dimensionality reduction. We do clustering. So this is what we do here. Actually, we have now clustered the pixels based on similar abundance profile across all these proteins. And so here are all the different pixel clusters. Here, a zoom in to the cortical region. And if we look at them, I mean, there's, for example, the apical surface comes out as a cluster, which is not a single cell, right? It's just a subcellular structure, which is nice to have as well. There's the dorsal radial glia, dividing cells, excitatory neurons, nuclei, and excitatory neuron processes, and so on. Actually, in this organoid, we also had then a general two clusters that were actually all the cells up here are dorsal telencephalon, all the cells down here are ventral telencephalon. We could actually see the pattern emergence over time if we then look across these organoids. We actually also got some primary tissue, and so this is the same antibody panel now on a primary tissue. I must say this is gestational week 13. It's way older than this, 8-week-old organoid. So it's not comparable, but it just shows where we are trying to go. So now in this case, I mean, it's of course way more beautifully structured, but it's also older. And now you can zoom into these different regions and look at these clusters. And eventually we would want to have time course data and bring that all together. Finally, one thing I want to mention is also spatial transcriptomics, here Murfish-based, so using the MERScope. You remember from our atlas, we found this stress signature, the metabolic stress. Actually, in this case, this is an organoid slice. This shows all the transcripts. Here it shows DAPI, you might not see it well. And here, I hope you can see this, you see this ring. And actually inside, DAPI is missing. These cells were actually dead. It was this necrotic core that we know. And this metabolically stressed cells are actually really at the border to the dead cells. So it's really a spatial signature. If the organoid gets too big, you start having this wave of metabolic signature that leads eventually, seems like, to the death of the cells. But it was cool to see now to actually spatially localize this signature. And with this, we are at the end. I have no idea how we are on time. But yeah, I think I don't have to say much about this. So yeah, I mean, I think we both see a bright future for AI combined with organoids and single-cell technologies. And here are the acknowledgements for both our groups. I think we both mentioned the people throughout the talk. Then, of course, also thanks to the funding sources and thanks to you.